Welcome to the Garden Talk podcast, where we interview growers from all over the world, both beginners and experts, seeking to learn more about what they know about gardening and how they do things in their garden. What's up, everybody? For those of you that don't know me, my name is Chris, aka Mr. Grow It, and you're tuned into the Garden Talk podcast. This is episode number 159. In this episode, I interview Colin Bell. He's a scientist who studies soil microbes and plant-microbial interactions. He completed his Ph.D. in Soil Microbial Ecology in 2009 and is well known as the inventor of Mammoth P, a popular microbial inoculant. He currently works for Microbial Mass, a microproduct that helps promote plant vigor and increase biomass. This episode, we focus on microbes, of course, everything from the type of microbes, how they function, how they can be applied in the garden, and more. If you want to see short clips of these podcast episodes, search Garden Talk Clips on YouTube. I also have a gardening channel where I show the plants that I grow. Both of those channels will be linked down in the YouTube description section below. One of my goals for this podcast is to bring free information about gardening to the general public. That being said, I'd like to thank the sponsors of today's episode who helped make that goal possible. Thanks to AC Infinity for sponsoring this episode. Check out their all-in-one pH pen, which measures pH, EC, TDS, and temperature. The sensor probe is replaceable, and it comes with storage solution and calibration solutions. Click the link in the description section below so you can learn more about their all-in-one pH pen, and the discount code MrGrowIt15 works on both Amazon and their website, acinfinity.com. Stash Blend. Stash Blend consists of several ingredients in one, so you no longer have to buy individual bottles. It includes corn steep liquor, seaweed extract, humic acid, beneficial bacteria, silica, and mycorrhizal fungi. It can be used in addition to your synthetic base nutrients or be used in living soil systems. Simply mix it into water and water your plants with it. You can get it at stashblend.com, link in the description section below, and use the discount code THESTASH. Spider Farmer. Check out their G series of LED grow lights, which have recently been updated to add some new features. With Smart App Control, you can set programmed schedules, adjust brightness levels, and simulate sunrise, sunset. The G-Series lights, except the G3000, feature a foldable design, requiring no assembly, making it ready to hang straight out of the box. More information is linked in the description section below, and discount code MrGrowIt5 works on both Amazon and their website, spider-farmer.com. And we are back, and I'm joined with Colin Bell. How you doing today?
I'm awesome. How are you, Chris?
Doing good, man. Thanks for coming on to the podcast today. We actually I ran into you at MJ BizCon, and kind of put you on the spot a little bit. I had a live stream going. I was like, there's Colin Bell over there. Should we talk to him? And then came over to you. You were nice enough to be on the live stream. You answered a couple questions, and I invited you onto the podcast, and you accepted. So put you on the spot. Appreciate you being okay with that. To be honest with you, you have been on my list of potential guests for years, and I finally have been able to get you on here. So really excited for this talk today. We're going to talk all about microbes, which is your specialty. But before we get too deep into it, can you tell us a little bit about yourself and how you got into gardening?
Yeah, absolutely. And again, it's a pleasure. It was a pleasure running into you, and a lot of fun being on the show. I've been following it for years, since 2020. And so it's good stuff. For me, people that don't know me, my background professionally is a scientist. I have a Ph.D. in microbiology, and I've always been fascinated with nature and natural processes. And at some point, as a young adult, I wanted to have more purpose. I engaged in nature and did the hikes and all the things I love to do. And that drew me into the biological sciences and ecology, and then went on this pathway to become a soil microbial ecologist. And ultimately, going through a master's and then getting my PhD in that area. And so it's been a heck of a journey, and that's led to a lot of really fruitful professional experiences also. And I think why I love soil so much is because as a very early age, and my parents, we grew up in West Texas. They were from North, but then they were both academics, and they both landed jobs at Texas Tech University in Lubbock, Texas, where I grew up. And they were fanatic, if not obsessive and compulsive around gardening. They spent all their free time changing every bit of yard into a different garden bed. And they had me in the yard with my little shovel, and my shovels got bigger as I grew, digging holes, helping to cultivate the soil. And so I do believe that, soil and working in the soil and growing plants is honestly something that I just grew up with. It's in my DNA, so to speak. And I can't help myself. Every time I move somewhere, and I've moved several times across my life, I've always started with the property and transformed it. It's almost a form of art for me, creating perennial beds and garden beds and flowering beds and annuals. And so my kids think I'm very obsessive in gardening and working in the yard. I spend all my free time working in the soil out in the yard here. And so yeah, I'll blame my parents, but it's just a part of who I am. So I get a lot of enjoyment out of gardening and just working in soil, and I'll just leave it at understanding at a deeper level from all my academic training, processes and soil processes. Just make it that much more, I think, intellectually stimulating for me. I just think about, and my mind's going the whole time, and I'll just go into these huge intellectual or mental rabbit holes, so to speak, and just kind of have these fun daydreaming experiences while I'm doing physical activity in the yard.
That's awesome. And I know one of the things you're most known for is you are the inventor of Mammoth P. And I got to imagine that there are a lot of people in the audience right now tuning in that use Mammoth P. So you are the inventor of Mammoth P, and then you switched over, and now you're at Microbial Mass. Is that correct?
Yeah, that's exactly correct. And that journey quickly working at Colorado State University after I graduated, I did a lot of professional gigs at the United States Forest Service and the United States Department of Agriculture Research Services, and that was between West Texas and here in Colorado, where I live now in Fort Collins, Colorado. Ultimately, I plugged into my dream location professionally, which was the Natural Resource Ecology Lab at Colorado State University. And we were such a driven group. It was very, very stimulating. All my academic heroes were housed at the NREL, Natural Resource Ecology Lab. And after about 4.5 years at that facility, some of my peers and colleagues, we wanted to make a difference quicker than just the scientific process of writing papers and getting grant and going down that academic setting. And we started thinking about and challenging how we could be more applied so we could get solutions for farmers now instead of, that someday someone will transform our science into application approach. We realized we had to take it upon ourselves. And so we started developing microbial natural solutions that were solving real world agriculture challenges that we had identified. And that was the journey of creating, ultimately it's known as Mammoth P, and what started the track of developing the company Mammoth, which was a great experience. It was about an 8-year run for me before I did leave, as you mentioned, in late 2022, just to start bridging out and working with other teams and other technologies. And so this venture with MIM Horticulture and the Microbial Mass group has been really rewarding.
That's awesome. Yeah, I remember when Mammoth P kind of hit the market. You refreshed my memory on what year it was. I believe it was 2017 or something like that.
Well, listen, I left the university in March 16th, 2015 to start the company. We had a little bit of money, no resources, no facility, started in a 1-car garage, and just started developing a little biofermentation facility, literally in a little 1-car garage in a rental flat me and my buddy had. And that quickly scaled through the last half of 2015. I got my first customer. So the thing was, I left the university as the co-founder, the operational co-founder, to start the company. 0 team members, just me. And my call to action was I had to scale up technology, build out a team, commercialize, go to market, and start making revenue. And we had about 6 months to do that before we ran out of money. And so there was a lot of quick moving. Ultimately, I remember having my first sale in Denver, and we were here, so we were hitting the Denver market pretty hard in August 16th. my first $100 bill, and I thought, wow, as an entrepreneur that had an idea, and we converted this idea into a actual living product that people could use in their gardens was a fascinating journey.
It's been a game changer in a lot of gardens. Microbial inoculants in general can be very, very beneficial in the garden. As we continue on here talking about microbes, I want to warn you that we have a wide audience here. We have beginners, intermediate, and advanced. And some of these questions are going to be for everybody, right? I like to start with the basics, the beginning. Can you start by explaining what beneficial microorganisms are and how they function in the garden ecosystem?
Sure, absolutely. So, what I would say is in general, most microbes, especially the beneficial microbes that help plants grow. When we think about beneficial, there's actually beneficial microbes for humans, and we call them probiotics. And so that's a really nice, familiar way to start thinking about beneficial microbes. For plants, most of them live in the soil, and they have exchanged value and help support plant growth since they've existed since the very beginning of time. And they do that by assisting plants in several different ways, by engaging with them, living in that habitat of their roots. And they can do a lot of things. They can help stimulate some levels of chemical signaling, which allows plants to elongate their roots so they're able to take up more water, more nutrients. What microbes are very well known for in natural settings and in farming settings alike is nutrient cycling. So nutrients can get bound up in substrate and soil and become hard for plants to take them up. Well, microbes do a great job of exuding enzymes and other chemistry that helps continue to create bioavailable nutrients, which plants need to support their growth. And so we know all the macro and micronutrients, and maybe we're not that familiar with it, but basically, just like humans, plants need certain vitamins and minerals and elements. We're all made of carbon and nitrogen and all these specific elements. All of them are listed on the elemental table. There's a select group of macro and micronutrients that plants need. The only one that they really get from the air is carbon, and everything else they get from the soil. And some of them are very hard to extract from a plant perspective. Plants do a good job of drying up, but nutrients can be bound and hard to liberate. That's where microbes play a critical role in natural ecosystems, cycling nutrients and allowing plants to continue to take them up so they can grow and be successful in natural settings.
Got it. And what type of beneficial microbes should gardeners be aware of, and kind of what roles do those play in supporting plant health?
Yeah, that's a great question. So there's a wide range of microbial applications that gardeners utilize, whether you know it or not. If you're growing in soil, there's microbes in that soil, and the plants are taking advantage of that. And just in a natural setting, there's no plant that exists in nature or anything in soil that doesn't have a microbial association. A lot of home gardeners and even professional gardeners alike like to engage in composting activity, where there's a lot of material and green waste that can get decomposed over time and then ultimately applied to systems where it's nutrient rich and microbial rich. And so those are what I would call some very general applications where there's a wide range of undefined general microbes that we know are going to support plant growth and liberate nutrients, and they're naturally occurring. There's other groups of microbes that are more of that precision application that you can actually purchase and do research, and they're manufacturers. It's actually a huge agriculture industry. That's a biostimulant industry where different companies manufacture or ferment or create different microbial formulas to perform specific functions. Some of these formulas might engage the plant to stimulate rooting. Others might engage the plant to stimulate different nutrient cycling. Some microbes are actually applied just to perform a pesticide function, where it's going to alleviate plants from certain pest pressure. What I'm familiar with from a manufacturing and scientific point of view is developing microbial solutions that actually liberate specific elements like phosphorus and calcium and iron, which are selected because they're a little stickier chemically, which I mean they're harder for plants to maximize the uptake of those specific elements in natural systems in particular. And so that was the motivation behind the science that the team I was associated with at Colorado State University was targeting to solve that ecological problem, the phosphorus problem. And there's still a phosphorus problem in agriculture, and there's still companies looking to solve that particular problem.
I heard a crazy stat. I forget where I heard it, and I don't even know if it's true or not. You'll be able to tell me if it's true or not. But I heard that 99.9% of microbes haven't even been identified yet. Is that true?
That's what they say. What if you haven't identified them, you can't tell for sure what that exact number is. There's at least 10x to 99x microbes that are very hard to identify. And I'll tell you a pretty fun story thinking about that and my journey manufacturing a product, Mammoth P. What we wanted to do was collect all the microbial diversity we could from nature. And so we collected soils in desert systems and overseas and in agriculture systems and forested systems. And we grew up all the microbes we possibly could. And, the environment selects. And for microorganisms, the environment to a large degree, it could be abiotic, meaning a temperature or a moisture, or maybe a chemical pH type of environment, which is helping to select their success, meaning will they persist and grow well or not. But to a large degree, the food source is a huge selective criteria. And so you could take the same soil samples and have two very different food sources and actually nurture and grow very different microbial communities and community assemblages based on that food source, which is fascinating. And so not only is the food source, but the time of growth. And so you think about growing microbes, and the microbes I work with will grow in a couple of days. They'll be fully mature and have an exponential growth curve going in just a couple of days. Some microbes take weeks to grow. Some microbes take months to grow. And so the persistence, the time is a factor that really predicates the complications, not only the diverse food sources, but the temporal or time that it takes to actually nurture some of these microbes are all reasons why it's very hard to extract most of the microbes from nature.
Talk about food sources. It reminds me of folks that use molasses. It's something I used to use a lot. I know people use it every single week. They're adding in molasses. And what I've heard is that if you continue to use that as a food source, you'll have some microbes that feed off of it. Others, not all, feed off of it. And you could be throwing off the balance of the soil food web if you just continue to add molasses in there. Is that true, or is that just bro science?
Well, actually, that's my exact perspective, and it's not just it's not just from my knowledge around select food sources. Like I have microbial media books, and they're about that thick, and all these different formulas with different carbon substrates and these different types of chemical agents select for specific microbes. And so I know for a fact there's a specificity that different media and sugars nurture in microbial communities. And there is a lot of great papers that are published in the scientific literature that shows exactly what you just shared, where if you feed molasses, and it wasn't even for that long term of a study, it will allow the microbes that grow and can eat or consume molasses very well to grow very well. And the others that don't grow so well or can't utilize that substrate, that sugar so quickly, they won't grow as well. And what you find, and this is a way that I think about it, life and especially microbe life, it's a probability game. And so once you get to a certain threshold or mass as a species, you have a competitive advantage. You're actually taking up space and occupying that space, which means you're occupying the supply, food supply in that space. And so if a food source can bias microbial growth to certain species, then it's also going to predicate the community structure, just like you said.
Wow, that's really interesting. So would you say that molasses should not be used in the garden at all, or are you for it just once in a while?
I don't like to tell people what to do. People use molasses, they have success, and so that's not the position I would take. What I would say is, I say this consistently, plants feed microbes. It's called exudation. And so from the beginning of time, and molasses is a sugar, and a sugar is a carbon, chains of carbon. Well, the plant gets carbon as CO2 from the air. It cleaves off the oxygen and continues to consume that carbon. And it creates chains of carbons. And ultimately, we're all made of carbon. We're mostly organic, means that carbon backbone. We're mostly carbon. Same for plants, same for microbes. What plants have evolved to do from a long time ago, and the ones that have persisted and succeeded. It succeeded in biology means able to pass their genome to subsequent generations. That's success. Creating a seed and having that seed propagate or vegetate. That's success in biology. But the successful plants that have evolved over time learned that if they consume that CO2 and transform some of that into simple sugars and exude that in the soil. And there's all sorts of varieties of exudate. The microbes will feed on that exudate. They'll grow to these masses and they'll cycle the nutrients. And there's a relationship and an exchange of carbon for different nutrient cycling and soil functions that the plants actually engineer through rhizosphere priming or through exudation. So the priming or that exudation isn't specific. It changes over time, and that chemistry can change by species. That chemistry can change by plant needs. And so in some ways, I feel like the plants are actually directing the traffic below ground, and they can exude different chemical species and different types of sugars over time, and slight varieties, including some amino acids that they'll trickle in there as well to nurture the community, the microbes, feed them, that support them. And so that's a very interesting way to think about feeding microbes is, why do we think we know what's best? The plant knows better, and the plant's going to do it naturally. And so I like to leave that function to the plants.
It really is incredible to know just a small piece of what goes on in the soil, in that ecosystem, and how it works. It can be mind-blowing. I want to get deeper into bacteria. And this is where I kind of ask you a broad question, and we'll see which way you run with it. Is what are some important species of bacteria that we should be aware of?
Well, listen, I'll be very general, and I'm not as biased about species as maybe some are. And the reason is because I've looked at tens of thousands of species and characterized them physically, meaning not only physically but functionally, physiologically. So if you can look at, let's just say 1,000 different bacteria that we can collect from soil, and there's a bunch of different groups. If we know there's family, then there's genre, then there's species. Microbes have functional redundancies. And so that's a really important characteristic. Microbes don't have a specific function. They're biology, just like every other biological organism, and whatever they do is going to overlap. If we talk about cycling nitrogen or phosphorus or, chelating or exuding some chemistry that's going to initiate root growth, most microbes have some competency within that range, which is fascinating to think. And I've shown it, and we've collected data where if you look at a wide range, let's say 1,000 microbes, they all have some level of competency, but some do it better than others. So thinking about that functionally is interesting, and that's why you think that there's a wide range of microbes in soil that persist and support plant growth. The interesting point is the resistance and resilience of those particular groups, meaning if a lot of different groups do the same thing, which ones are actually going to live and do things well. And so I used to do a lot of work in desert systems. And what we saw was a ton of functional redundancy, meaning a lot of the microbes did the same thing. But what they had was very different environmental tolerances. Meaning some microbes did very good in high rain events, while others didn't do great in those events. And most microbes in biology like water, but they did great in dry, hot events. And so you wanted that diversity because you need soil function across that wide range of environmental variability. So what you'll find in most, let's just say products, bottled microbes that you can access commercially, you'll find Bacillus genus, and there's a whole array of different Bacillus species. The reason you find them in agriculture is because of their resilience in environmental conditions. Bacillus species are dormant species. They actually create things called endospores, which is the dormant state of their vegetative cells, meaning they can go in this dormant state. You think about plants, a seed is a dormant state of a plant. If you think about fungi, spores are what's called this, it's like a seed, but a dormant stage. Well, bacillus are one of the only microbial bacterial genres that produce that type of dormant state, and it's called an endospore. And the reason you find mostly bacillus species is not necessarily because of their function, because there's a pretty good overlap of function between these bacillus species, but they're very resilient, meaning that they can persist for a very long time, especially in dry environments in a dry carrier. They can persist for years and years in soil in very harsh conditions. And then when the environment's right, they'll vegetate, which means activate, and do what they do to help support plant growth. So I would say by far the majority of bacterial species you're going to find in any type of agriculture or gardening application is going to be different bacillus genus.
I'm familiar with a handful of them. I know Bacillus subtilis is kind of the most common. It seems like it's all over our skin, apparently, right? It's like the most common one on our skin. And
100%. Well, listen, Bacillus subtilis too, it doesn't do just one thing. Even this product, Microbial Mass by Mammoth Horticulture, has many bacillus species in there. And the scientists that developed that particular technology made sure that they could identify some functions of these different species that they did very well. But Bacillus subtilis does many functions very well, which is interesting. And because they can be dormant for long periods of time, they can be applied into the market, into agriculture settings without dying. And that's important. You can have the best, and I think that's the difference between academic, fundamental science and applied science. Academics get caught up, and I'm not speaking derogatorily. I was an academic, but I got caught up on looking at something that was super cool, and I absolutely fell in love with it on the lab bench. But if it doesn't have the other applications and features of persistence and able to be compatible in different environments for different applications, it becomes very tricky to use that type of organism outside of just enjoying its coolness.
Got it. Now you mentioned dormant, and I just want to kind of get a little bit deeper into dormancy. From my understanding, microbes can go dormant. and they can get out of that. For example, if you let the medium dry out too much, then my understanding that the microbes go dormant. But when you re-moisturize that medium, they wake up. And dormancy, for those that aren't familiar with it, in layman's terms, it would say a sleep state, right? Is that all true? Is that do any of the microbes kind of die off due to drought?
They will. And so again, it's a probability game. There's never an always in biology, and there's never a never. And so there's always a range of what happens. And so, let's talk about the two contrasting groups of microbes. I worked with microbes, and in the IP, the patent that I developed for Mammoth P, they were not dormant bacteria. They were rod-shaped, not able to form endospores, but they were very robust, and they had a really interesting membrane, which allowed them to just hold their shape, even in very dry conditions, in very salty, high ion conditions, and high osmotic pressure. No, these microbes came from soil. They're not used to being in a fertilizer tank. And so we had to select the ones that did do a good job. And it turns out that, these microbes in that particular product were able just to maintain their structure, even if they weren't functioning very well and they were just holding on. Once the conditions became favorable and they got inoculated or they collided with the root zone, that's their habitat, that's home, and they could start really functioning and rocking, with what they do. So bacillus in particular have evolved to immediately flip from an active state to an endospore state or a dormant seed state, like hibernating, but it really transforms the structure. And I guess the lesson that it's basically a law, but what it is structure equals function. So what happens in some cases with high osmotic pressure, a lot of water, or extreme drought conditions, is it changes the water availability or the ability of the microbes to regulate their osmotic pressure within the cell. And if they can't regulate in a very dry environment, hold on to their water, or in a very wet environment where they have to continue to exude water because they'll take in too much water ultimately. And they're cells. So think about a little circle, or think about a balloon, but microscopic so you couldn't see it. Think about a water balloon. It holds its structure until it doesn't. And if you have one little pinprick, what happens? The insides squirt out, and that water balloon no longer holds water. Well, that's what happens with microbes across the board. If they can't figure out how to maintain that structure to live to fight another day, they're going to exude. It's called lysing. Their cells will lyse, and their insides will just release into their environment, which becomes a food source. It's like little vitamin packs, so it's not necessarily a bad thing. But so some microbes will lyse during these extreme conditions, depending especially how quick the shift is from dry to wet to dry, and others can maintain that environment relatively well. That also happens with pH shifts. So if there's more hydrogen ions or acids versus OH ions, well, microbes at the cellular level have to regulate that. So they have these pumps literally built into their membrane, to their skins, and it pumps out to balance the outside with the inside. That's what microbes are always doing. And it turns out that a lot of the microbes that are very environmentally robust are better at pumping. They're better at regulating that osmotic pressure, that ion concentrations from their interior to their environment.
Got it. Now, what about like powdered versus liquid form on these microbial inoculants? It kind of reminds me of that. I got to imagine that some of these powdered forms of microbes, they just have those microbes in there that can last in a dry form. Is that right? Can you talk to us about the differences between liquid and powder microbial inoculants?
Yeah, that's right. And so that is the exact difference. There's many different carriers of a powder. Let's just talk about that for 1 second. Every one of them are going to be microbes that can form a dormant endospore. So it's going to be very hard for you to find any dry powder microbial solution that isn't a bacillus or a different arrangement of bacillus. And all of those are not active in that bag. They're all in that endospore form. And so they can stay there for a very long time, which makes it effective for the manufacturer. Once those microbes are introduced into an environment that they sense a food source, they'll vegetate. It's what it calls between a dormant state and a vegetative state. They'll activate and start doing what they do, but they're sitting there in a seed form, and all it is protein, just like a seed, with some DNA and a little bit of sugar, carbon, that when they can sense the environment, they start regulating the DNA, creating the proteins they need to, and turning into a completely different form.
That's incredible. Yeah, I was always curious about liquid versus powder and kind of the differences there because I know some manufacturers can't offer a powder, although there are some benefits, shipping wise and stuff like that, having a powder versus liquid. But I was just curious on that one.
Well, I'll tell you, with the Mammoth P product, I wanted to have a powder so bad, but the microbes in Mammoth P, I could never find a carrier that I could get them to persist well in. And so I could never get recovery when trying to impregnate different dry powder solutions with the Mammoth P microbes. That's why we only had a liquid offering. There are Bacillus endospore related bacterial products that all of them are dry, and some of them are actually also offered in liquid form. And so it really depends on the microbe and how much elasticity you have with that particular group.
Understood. One of the most interesting, in my opinion, bacteria out there are nitrogen-fixing bacteria. So microbes that can actually take nitrogen from the air, convert it, and put it into the medium form that the plant can then uptake. Can you talk to us about microbes that fix nitrogen from there?
Yeah, well, there's a really specialized group that have done that, and that's what's so cool about these. It's science, but once you start getting into it, you're like, it might as well be science fiction because it sounds like a science fiction book, the way they do these things. But there's microbes that can persist in very high salt contents or very hot environments or very, very sulfury environments and all these different environmental gradients. There's not as many groups as just the general groups that persist within a range of temperatures and relatively normal ecosystems. But the azobacteria group, they're fascinating, and what they can do is they're not necessarily always fixing nitrogen, but they're able to do that and pull nitrogen, which is a major element from the air. I don't know how many people realize that, but we're breathing in nitrogen all the time as far as N gas form, which is it sounds counterintuitive. These microbes have evolved to take advantage of that, where they weren't very able in certain conditions to persist because of low oxygen. So they create these different rhizomes where they can embed inside these plants, and it creates a habitat that they're very comfortable in. That's, an evolutionary strategy that allowed these particular groups, for whatever reason, to succeed. And it was a successful strategy because they're still around today. And what they're able to do is, in that very, very low oxygen environment, pull in nitrogen and use that as an electron acceptor for respiration. And so that's the mechanism they're trying to survive. Microbes breathe just like we do. They have to take in oxygen and they take out CO2. But microbes have evolved to be able to adapt that strategy to very, very low oxygen concentrations or dissolved oxygen concentrations. And in some cases, there's groups of anaerobic, which is no oxygen environment bacteria in particular. And so when there's no oxygen and it's anaerobic, you can't take in oxygen. There's none to have. And so you can't facilitate respiration in that electron transport chain to live. And so they've adapted these strategies to pull other elements that they're able to pull to facilitate respiration in those particular environments. So it's an amazing group of microbes. You'll find them in agriculture quite readily. And what they do is they associate, especially in rhizome related conditions, and they're able to help feed the plants nitrogen from air, which, pretty cool.
Yeah, it's helpful instead of having an input to get nitrogen in there, whether it be organic fertilizer or synthetic nutrients, whatever. Adding some beneficial microbes in there that can actually grab it from the air. That's huge. That's a big key, and I know it is the azos bacteria that's in a lot of microbial inoculants.
Yeah, totally. And I'll tell you, that's a strategy for a lot of crop rotations. You talk about farming rotations where you're trying to keep plants in the ground year round. So you'll have a cash crop and you'll have what's called a cover crop. And sometimes farmers will plant different crops that actually promote azo nitrogen fixing bacteria in the soil, and it replenishes the soil in the off season with nitrogen from growing that cover crop. Super cool strategy.
Oh, that's incredible.
Yeah.
Another type of bacteria that I feel like is being talked about more and more these days, and it's very interesting as well, as endophytic bacteria, endophytes. Can you talk to us about endophytic bacteria?
Well, I'll tell you what they are, and I think there's a lot of nascency to understand exactly where they play, but endophytic bacteria, and there's fungi that do this also, are bacteria that actually incorporate inside the plant. And so they'll work between the cells of the plant cells and work their way and grow within the plant. And there's always an exchange, is what I'd say. I've heard more and more groups trying to develop endophytic bacteria for either, from a nitrogen fixing point of view, or maybe some other poor views, but there's always a trade-off. And so if microbes are growing in the plant's giving it something to incentivize it for something in return. I would say another, even a more intuitive example, because, the jury's still out on exactly how beneficial endophytic bacteria are in application. I think that there's a lot of great science. I think it's a little on the nascent side. I've heard it ebb and flow for a long time, and I think that there's really fun applications. I just don't understand where there's a compelling product that I've seen a clear mode of action for a long time. But I will tell you something that's been around for a long time, and it's in the fungal category. It's arbuscular mycorrhizal fungi. And so I know you know about this. Everyone knows about AM fungi, but it's the same concept where these fungi actually infect and grow inside the plant root. Part of them live inside the plant. That's the only way they can be successful. And there's an exchange for these arbuscular mycorrhizal fungi, where they'll create these sites of exchange within the plant root. The plant has to give up something, and it gives up carbon. The plant's always giving up carbon. The plant's always giving up sugar. It's a charismatic carbon pump. It can look like anything it wants to look like, depending on the species, but it's drawing in carbon, and it's transforming some of that into sugars to feed the soil and to feed the microbes. The arbuscular mycorrhizal fungi in particular, called AM fungi or other things, are actually scavengers. They actually have part of their bodies filamentous hair-like structures that exude into the soil past the root zone, and they can scavenge available nutrients. And what they do a great job of is scavenging phosphorus. And so they're able to draw that phosphorus from beyond the root growth zone and bring it back to the plant. And in these arbuscular sites, they exchange phosphate to the plant for carbon from the plant. And so, although it's not endophytic bacteria, that's an endophytic relationship. It's a clear symbiotic relationship that I think emulates a very, very clear mode of action with that same type of concept.
So when you see these mycorrhizal fungi products that market unlocking phosphorus. Is that really what they're doing? Is it just an extension of the root zone, and that's how they're accessing that phosphorus?
That's right. And so, and that's a little bit of a misnomer. Arbuscular mycorrhizal fungi, to unlock phosphorus, typically you're talking about a balanced source of phosphorus, and you're transforming that from a plant unavailable form to a plant available form. That's what I would call unlocking phosphorus. But, let's not get into semantics here. What I would say is arbuscular mycorrhizae are not very enzymatically capable. So they're not doing a lot of the unlocking. They're more like scavengers. They're kind of like the hyena of the particular fungal kingdom. They're able to infect the plant root. They have a transfer location within that plant root, within the cortical cells. They develop these vesicle arbuscular sites, and then they extend out. And to be honest with you, bacteria are much more enzymatically capable to liberate phosphorus in these micronutrients. And so the fungi can grow and, they cohabitate. There's, tens of thousands of bacterial and different fungi within, very, very small, even grams of soil. And so these fungi can go, and as they sense phosphate that's in a bioavailable or soluble form, they can suck it up very competently. And then they translocate it. They keep some of it for themselves, and they translocate quite a bit of it to the plant because they like that sugar the plant feeds them. So they're scavenging phosphorus, and you can consider that unlocking phosphorus or grabbing it from the soil. The plant didn't have it, so they are through that sense. But that's something that's a little bit of a misconception. They're not necessarily liberating; they're capturing something that's already been liberated. And I'll say something else about phosphorus or mycorrhizae that's a little bit of a misconception. And there's a great study, so go look at some of these papers. We can probably provide some in the links, or it's publicly available information. The higher the phosphorus availability for plant, the lower the infection success rate is for mycorrhizal fungi. So if phosphorus is available, the plants don't let them in as easy, is what that comes down to, which I think it really speaks to the environmental sensitivity of the plant. The plant doesn't have to give up that carbon if it doesn't want to. And if it's getting what it needs from that particular microbial function, it won't let them in. And so the infection rate of mycorrhizal fungi in a high phosphorus environment is much less than the infection rate of mycorrhizal fungi to a plant in a very low phosphorus environment where the plant actually needs that symbiotic relationship.
Wow, that's interesting. Yeah, I thought with unlocking phosphorus that it was actually like the fungi was doing some sort of nutrient cycling and then turning it from a unavailable form to an available form. But that's funny. That's just an extension of it and just kind of grabbing it where the roots really can't grab it.
Yeah. Look, there's other microbial fungal groups and obviously bacteria that do a great job of that. And that's why, they coexist, but they wouldn't be as competent if they didn't have the other microbial contributions within that soil environment to be able to scavenge that phosphorus.
Okay. Endomycorrhizae and then Ectomycorrhizae. I kind of want to get into that a little bit because I feel like a lot of my audience doesn't really know the difference. I think we often hear that one of those isn't actually usable by medicinal plants. Can you talk to us about the differences between endo and ecto?
Sure. So endo is related to the Arbuscular mycorrhizae group that we just talked about. And so there's many different species of endomycorrhizal fungi, and there's a lot of specificity. And so there's, if you talk about the genus, species, just getting scientific, just to put a name to it, it used to be called Glomus introdecies. You can also see it as Rhizophagus introdecies. It's the same exact thing. Mycologists or scientists that study fungi love to change names for whatever reason. And so there are certain species of fungi that do pretty good with medicinal plants, but there's only a couple that are actually very relevant to my knowledge. But there are other arbuscular mycorrhizal species that do a very good job of interacting with other different plant species. So there's a lot of plant specificity with the endo or Arbuscular mycorrhizal fungal group. The ecto, the same way, and they're more associated with a woodier, shrubbier type of plant. And ectomycorrhizal fungi don't penetrate, but they grow like a net or a sheath around the root zone. It's called a Hartig net for whatever reason, because the scientists actually that first discovered this, I think it was, I forget his first name, but it was Dr. Hartig, if I'm not mistaken. I could be completely misremembering that, but I think so. This is old school stuff, but it's just literally a layer of fungal mat that grows around and covers the outside perimeter of roots. And so that's ecto. It creates a lot of benefits also. It creates a very, very highly functional and enzymatically capable area right around the root zone to maximize nutrient uptake. And so Endomycorrhizae or mycorrhizal fungi are a very different group of fungi than ectomycorrhizal fungi. Ectomycorrhizal fungi are typically very highly functional, very enzymatically capable, and will do a good job of carbon and nutrient cycling to a lesser degree. They do a lot. These ectomycorrhizal saprophytic fungi, free-living fungi that actually also produce a lot of these ectomycorrhizal associations. And bacteria and the mineralizers, these bacterial as mineralizers also interact with this ectomycorrhizal layer. It creates this crazy habitat, a hotbed of nutrient cycling or nutrient availability. And so it's a true benefit to plant growth in particular.
So ectomycorrhizae can be beneficial to plants like hemp?
No.
No.
They're not. No, that's not my understanding. Yeah.
Okay.
They don't associate. They do associate with different shrubby types of plants, and in some and some trees, and in some cases some grasses. But it's a very different group, and I don't know any ectomycorrhizal associations. Not that different medicinal or hemp plants can't benefit from free living fungi, because they are very enzymatically capable and they'll stimulate some nutrient cycling, but they won't form that true structure.
Okay, got it. Just wanted to get clarification on that one. Appreciate it.
Yeah, sure.
Colony forming units, CFU. So when somebody picks up a bottle of microbial inoculant, for example, they look at the back, CFU is one of the things that are mentioned on that. Can you break it down? What is CFU? And I know there's not going to be a clear-cut answer to this, but what's an appropriate amount for bacteria, fungi, so on and so forth?
Yeah, I'll just go. CFU is just a measure of density. And so why it's a colony forming unit, a colony is a visible metric. And so bacteria and fungi on the very early stage growth scale are invisible. They're microbiology. Micro means naked to the, or not visible to the naked eye. It needs magnification. And so you think about a bacterial cell, well, you could never see it with the naked eye. A colony forming unit is when enough of these cells have reproduced, and bacteria reproduce clonally. They grow to a certain size, then they get too big to be efficient because the cell membrane is not at the right ratio for their internal components. And so they bud and they reproduce. And so they'll continue to do that. And they'll clonally reproduce, and they can do it very quickly at a certain stage. And once they've done that enough, a certain species where you can actually visibly see, quite honestly, it looks like a little blob on a petri dish. That's called a colony forming unit. The only reason that's relevant is because that's the measure, and it would be impossible to count the amount of cells within, let's just say there's 1,000 colony forming units. Well, there could be a billion cells that make up that 1,000 colony forming units. And so it's just a measure that allows us to quantify at some level some density of microbes within a particular solution on a plate, whatever you might think.
Okay, got it. I have a question that relates to hydrogen peroxide. So can microbial inoculants be used with oxidizing agents such as hydrogen peroxide or hypochlorous acid? will it kill off those microbes? I think that's a big concern for a lot of gardeners. They are using hydrogen peroxide in order to kind of sterilize things and prevent fungal root pathogens, for example. But they also want to use the microbial inoculants. They also want the benefits from microbes. Can they be used together?
Yeah, short answer, yeah. And I know that because I've worked with them for long enough. And so I'll qualify, as a Ph.D. microbiologist, I have to be really good at two things. One is understanding how to grow and propagate microbes, and the other is to be excellent at killing them, because I have to keep constant sterile environments and grow the microbes I want and not grow the other ones. And so really, really familiar with disinfectants is what we're talking about, these disinfecting agents. There's antiseptics, which we use on our living skin, like alcohol as an example, and hydrogen peroxide is actually considered antiseptic to a large degree. It's also used as a disinfectant in cultivation practices. The disinfectants are typically the bleach, hypochlorous acid, peracetic acid, H2O2 is an example of something that's been used. And in general, to kill microbes, you need two things. First of all, you need an agent, a disinfecting agent that can either create a static or a cidal property. Static means freezing growth. Cidal means killing growth. And so those are the two properties that qualify as a appropriate disinfectant. And there's a concentration and a time required on top of that. And so I've tested a ton of disinfectants trying to kill microbes just to see what it would take to kill them based on recommended applications. And there are very, very few agents or disinfectant agents in cultivation that are recommended for the appropriate time and concentration to effectively kill many microbial solutions. And it's because it's not concentrated enough and the time is too ephemeral. It doesn't allow them to sit in that solution. And so I would recommend that it's, don't change a thing if you're having good success. Far be it from me to change, but make sure that you could integrate with confidence the biology you want to. I would say it's better practice to stagger a disinfectant application from a microbial just intuitively. I mean, I wouldn't want to mix bleach with my microbes and say, okay, have at it, go. A bleach agent or a hypochlorous acid or any type of chlorine, H2O2 is more so to mitigate risk, to maintain a level of cleanliness through the lines, through anything that might be that might represent a risk over time if it grows. Once microbes hit the rhizosphere zone, that habitat is so complex that they're buffered from much of that anyway. And so if you think about roots, and I don't know if anyone on the call or you have ever seen roots at the microscopic level. It's nice to see healthy roots just at the macro level, and they're just white, structures. And you're like, oh man, that's pretty. If you look at it under a microscope, it looks like the canyonlands of Utah. There is surface area that's engineered naturally that ebbs and flows, and there's just nothing but microscopic canyons that the microbes embed in. And so the surface area is inherent for maximum absorption, and that's what roots function are. The more surface area, the more it can absorb. So microscopically, these things are going every which way. So there's so much habitat, and there's not a penetration typically in applications for disinfectants to actually get into all that surface area. And so it's a little bit of a misnomer as far as I'm concerned that you can sterilize anything biologically, because you can't. You cannot sterilize a root zone. You cannot sterilize the air. You can't even sterilize a table. You can manage the concentration or the population or density of different microbes if you do that well enough. In a root zone, it's absolutely impossible to sticky sugar substance with a bunch of microbes in it, no matter how you start. What I would say is you can either select the microbes you want in there or let your environment select for you. And typically, if you can have a choice of putting beneficial microbes, knowingly putting beneficial microbes in that substrate, in that root zone, you're mitigating a lot more risk than just taking the chance that something from the environment's going to come in, see and occupy a space that's open, and infect, because not all microbes are beneficial, let's just say.
I have learned so much from this podcast episode. I have a feeling my audience has learned a lot as well. You have
Awesome.
just so much good knowledge here. I appreciate your time today. We're going to wrap things up here. Can you tell the listeners how they can find you and what you have upcoming in the future?
Yeah, absolutely. Hey, listen, we keep on spreading the good word. We're about nature tech and MIM horticulture. We're trying to, make it easy from an educational point of view and from an application point of view to understand and use things like biostimulants and growing. And so you can always find me in my personal Instagram, Colin W. Bell, COLIN W. Bell. Same thing for LinkedIn. There's a website, mimhorticulture.com. You can always learn more about the technology that I'm representing here. Great group based out of Canada. We've been in the US for a couple of years. And overall, reach out anytime. I'm happy to answer any questions. It has been an absolute pleasure to engage and speak with you today.
I'll definitely have a link to Colin's Instagram down in the YouTube description section below. And if you have any questions that relate to microbes, leave those down in the comments section as well. Maybe we can, if we get enough people demanding a part 2, maybe we can get Colin back on for another talk. I mean, we could sit here and talk for 4 or 5 more hours, I'm sure. I want to thank the Patreon folks. Patreon.com/Mr. Grow It. The folks that are being scrolled on the screen right now are in the producers tier. Special thanks to you. You keep this podcast going, and I can't thank you enough. Colin, once again, thanks so much for coming on. Really appreciate it. And I'll let you go. Hope you enjoy the rest of your day.
Likewise. Hey, thanks so much. Have a good one.
Peace out, everyone. Catch you in the next episode.